Our body makes use of different proteins to get a lot of work done. We need proteins to fulfill tasks such as taking care of communication, to shape molecules into forms that are more usable for the body, to build new structures, or to deliver nutrients and building materials to where they are needed. Every protein in your body has a specific job, and every single one of them is important to keep us going. A lot of proteins travel around your whole body, while some only travel through specific cells. When it comes to traveling around within a cell, a protein usually has to follow strict trafficking rules, similar to the traffic in our real world. Before the invention of microscopes and labels, we were not aware of these trafficking pathways. With many years of work, scientists around the world have found out that it all looks a bit like a road system, with highways, city and country roads, and many other types of streets. Unfortunately, we don't fully understand all the trafficking rules for proteins yet. Until now, we did not find good enough ways to light up these roads properly and long enough for us to get all the detail about protein movement that we would want. Historically, we have used different tracking devices to help us understand the pathways in the cell. We have installed lights onto the proteins to help us light up the pathways. Unfortunately, the lights tend to turn off before we get all the information that we would want. On top of that, installing these lights sometimes causes the proteins to behave differently than before. It would be very helpful for us if we are able to understand the trafficking rules in a lot more detail than we currently do. It might help us to make use of the road system to deliver medicine to exactly where it is needed within the cell to treat diseases in a much more directed manner. For this, rather than using lights that can turn off, currently we try to install stable tracking devices instead. In our case, the stable tracking label is a gold nanoparticle. At Cardiff University, we have a microscope that can reliably follow a gold nanoparticle as it travels through a living cell. It reveals a lot of detail that we were not able to see before. We call this four-wave mixing imaging. The microscope will help us find the missing details of how exactly the proteins move on the roads, whether there are some yet unknown checkpoints, or if the environment changes from location to location. My name is Nicole Slesiona, and I work on this project at the School of Biosciences at Cardiff University in a joint project between Watson and Bory Langbein Lab as part of the MUSIQ Marie Curie European Training Network.